Hi, today I'm going to show you how I genetically engineered E. coli bacteria to allow them to survive in usually non-livable conditions. The goal of this experiment is to engineer E. coli so that they can live in environments that they normally can't, specifically strep plates. Strep plates contain a molecule called streptomycin, and when it binds to ribosomes of E. coli, it shuts down the protein synthesis process. In other words, the molecule stops the bacteria from reproducing or doing processes that allow it to live and grow. The mutation that can be made to allow protein synthesis is very small. It's only a change in 3 base pairs, which is enough to allow the E. coli to grow and multiply in strep plates. For this to happen, a small mutation is made in the bacteria's RPSL gene that encodes for the highly conserved RPS12 protein that prevents streptomycin from binding to ribosomes, allowing the bacteria to grow and multiply in strep plates. The mutation that causes this phenomenon is only a couple base pairs. As a review, codons are transcribed from genes, which are translated into amino acid sequences. Chains of various amino acids fold in particular ways to create functionalized proteins which contribute to the various functions in a cell. The mutation in this scenario is a mutation in the RPSL gene, causing a change from a lysine codon to a threonine codon. In other words, a change from AAA to ACC in the gene's transcribed mRNA. Lysine is an amino acid that, when in a particular position, shuts down the ribosome in the E. coli. Threonine, on the other hand, multiplies protein production. By changing lysine to threonine, we allow E. coli to survive in strep plates. If the experiment is successful, we will have a colony of E. coli growing on the strep plates. The first step is to make the plates, where we will let the bacteria to grow on. There are 2 kinds of plates that we use in this experiment. The first is LB agar and the second is LB strep con Arab agar plates, or just strep plates. The first acts as a home for the bacteria. It provides a livable environment with the right pH level, limitless food, and everything for the bacteria to grow and multiply. It is a standard media for growing bacteria in labs. These plates will be used as a medium to grow E. coli bacteria. The second, as you know, contains streptomycin molecules that inhibit the bacteria from thriving and multiplying. These plates will be used at the end of the experiment to test whether the bacteria has been successfully edited. Before starting the experiment, I would like to give credit to the Odin for providing me with all the necessary equipment to conduct the experiment. You can buy a CRISPR kit from their website at www.the-odin.com. First, add 150 milliliters of water into a glass bottle with LB agar powder, which came with the kit provided. Shake the bottle vigorously and heat the bottle in the microwave for less than 30 seconds a number of times until the LB agar powder is completely dissolved. The liquid should be a pale clear liquid after dissolving. Be careful that the solution does not boil and that the lid is placed on top and not tightly closed. Do the same for the LB strep agar powder and then pour each of these solutions into 7 petri dishes, with a total of 14 petri dishes. Let the plates cool for about 30 minutes before putting the lid back on, then leave them to dry overnight or a couple hours works just fine. After that, flip the petri dishes upside down and then store them in the refrigerator. The next step is to grow a sample of E. coli. Add 100 microliters of sterile water into the provided DH5A tube. Shake the solution and pipette all 100 microliters of bacteria solution onto an LB agar plate. Use an inoculation loop to spread the E. coli around on the agar plate and let the bacteria grow at room temperature for 1 to 2 days until there is a visible whitish bacteria. We're going to make the Cas9 system to edit the bacteria. To summarize quickly how the basic CRISPR-Cas9 system works, the system is made of 3 components: a guide RNA, a Cas9 endonuclease enzyme, and a template DNA. The guide RNA corresponds to the target gene, or in this case, the RPSL gene. It binds to the Cas9 enzyme, leading it to the target DNA location. The Cas9 enzyme then makes a double-stranded break in the target DNA, allowing intracellular repair mechanisms to attempt putting the DNA back together. Through these repair mechanisms, an edit is made. One such mechanism is called HDR, or homology-directed repair, where a nucleic acid containing a mutation is provided to act as a template for these processes to make an edit. This is a very simplified way to explain how the system works. If you want an in-depth explanation, check out 2 articles that I've written and linked in the description. These components are stored in plasmid form. Cas9 and gRNA encoding genes are inserted into plasmids and then inserted into the cells later on in this experiment. Bacteria have a cell wall that is semi-permeable, not allowing our CRISPR system to pass through. This is a big problem, and to bypass this barrier, we use a special mixture called the bacterial transformation mix, which contains 20% polyethylene glycol, or PEG 8000, and calcium chloride, which shields the negative charge of plasmids, helping the nucleic acids enter the cell. It helps because the cell membrane and nucleic acids are negatively charged and they naturally repel each other. And when you try and shield the negative charge, it is easier for the CRISPR components to enter the cell and hopefully reach the nucleus where it can do its editing. Add 55 microliters of sterile water into the tubes containing Cas9 plasmids, gRNA plasmids, and template DNA. Gently shake the solutions and put the tubes aside. To make the competent cell mixture, grab a microcentrifuge tube, pipette 100 microliters of bacterial transformation mix into the tube. Then, using an inoculation loop, take some bacteria from the LB agar plates and add it to the tube. After that, pipette 10 microliters of each of the CRISPR components into the tube. Shake everything together until it is dissolved. Make sure there are no big clumps of E. coli sticking around. The final step is to allow the CRISPR components to enter the E. coli cells by bypassing the E. coli cell wall. We will be heat shocking the E. coli, which will break or make holes in its cell wall, allowing our CRISPR components to enter the cell. Take the competent cell mixture microcentrifuge tube and put it in the fridge for 30 minutes. Instead of that, I put it in ice and it worked out well. Then take it out of the cold environment and incubate it in very warm water for 30 seconds. By doing this, we are changing the environment temperature drastically. The next step is to add 1.5 milliliters of tap water into an LB medium microcentrifuge tube and shake it until the LB powder is dissolved. And then pipette 250 microliters of the solution into the competent cell mixture tube. This will aid the bacteria to grow more quickly. Incubate the competent cell mixture, giving it time to recover from the heat shock and allow the CRISPR system to take action. Finally, pipette all of the solution inside the competent cell mixture onto a strep plate to see if our experiment works. If the E. coli grew on your strep plate, it means that the experiment works. After a couple of days, this is how my experiment looked like. Through this video, you've learned the basics of how CRISPR works through a hands-on activity. Thank you for watching, subscribe, and I'll see you soon.